Somebody confirm you can hear me?
I can hear you.
Great. Let me look again. There we go. Boy, that's rather odd. I'm the same place almost for my Tuesday, Thursday classes, Monday, Wednesday. I thought we were behind on, oh, wait a minute. Today's Wednesday. That's why it's the same place. We are behind. All right. All right. Let me get my schedule up here. Let me share my screen. All right, you should see my screen now. Today is January 31st, and we should be finishing chapter 3 today. We're midway through it. In the lab today, I'm just going to log in for 15 minutes to answer questions. If you have any questions, I will be there during the start of the lab time. If you have no questions, you're not interested in listening to any questions, you do not need to log in for the lab today. Any questions about any of that? Let me also state that you may work together in groups. However, if you do work together in groups, you may not simply use your study buddy's wording. Everything you write must be in your own words. So if you turn in a homework assignment and you're using your study buddy's words, that is plagiarizing. The only way you could avoid that would be to put it in quotes and say where you got it from. That is not plagiarizing. So when you do work together, make sure everything is in your own words. Any questions about that? Let me reiterate that if you do plagiarize, then you'll get a 0 for that assignment. And I've got that in the syllabus. Oh, come on. Sorry, I'm not getting control of the mouse here. So, oh, there it is. Let me blow that down a little. I don't know why that mouse is so trouble. I went over this point before. On the first day of class. I'm not seeing it here. I'm not sure why my. There it is there. If I discover plagiarism, the use of another's work without citing credit, you will lose points and may score a 0 on that assignment. Students must complete both the plagiarism tutorial and the plagiarism quiz. That's good enough. All right. So sharing all quizzes and exam data would be considered cheating. If I discover that, you'll also get a 0 on the quiz or exam. Any questions about any of that? All right. Let's go ahead and scroll down. And let's go to chapter 3. So this is the last slide I talked about last time, and that was looking at the Graham procedure. And that after you stain the cells with safranin, the gram-positive cells will look purple, the gram-negative cells will look pink, and that pink is actually in the gram-positive cells, but our human eye cannot see it. Our human eye only looks at the deeper, stronger color. It doesn't see the pink. You could see the pink if you looked at the cells under the electron microscope. you'd see regions of pink in the cell and regions of purple. But our human eye can only detect the purple. You then can blot the slide dry. Let me demonstrate. So let's say this is our slide. Can you guys see this? Anyone? I'm going to assume you can see it. You get bibulous paper and it's very thick, almost like cardboard. And you rip it out and then you fold it usually. And then let's do it that way. And then you just tap the bibulous paper down on the slide. You do not rub the bibulous paper against the slide this way. That will pull off the cells. But you may tap it down and that would get rid of most of the water. It will get rid of enough water so that you could then put oil on the slide and observe your slide under the microscope. And that we call blot drying. So the basis of the gram staining. Gram-positive cells are purple after the procedure. Gram-negative cells are pink. What is the basis for the differential staining? It's related to differences in the cell wall structure. The gram-positive bacteria have a thick peptidoglycan layer. The gram-negative bacteria have a thinner peptidoglycan layer. Here we're looking at the cells. Let me blow this up. The gram-positive cell, with the yellow being the cell membrane, the purple being thick cell wall made mostly of peptidoglycan. And this is showing you only 3 layers of peptidoglycan. As I stated on the gram-positive cells, in reality, that's at least 30 layers of peptidoglycan and more likely to be 100 layers of peptidoglycan in the gram-positive cells. There are a few other molecules. We'll talk about them in a little bit, but it's mainly peptidoglycan as the main molecule in the cell wall of gram-positive cells. In the gram-negative cell, it's different. You have the yellow cell membrane there, and then you have a thin peptidoglycan layer here. And then on top, you have another lipid bilayer. The lipid bilayer, let me blow that up a little bit. That lipid bilayer is called the outer membrane of the cell wall. It is a part of the cell wall. It's the outer membrane of the cell wall. And we'll talk more about its construction in a little bit, but not now. It does have a thin peptidoglycan layer. Here it's only shown as 1 layer thick. In reality, it's at least 3 layers of peptidoglycan and may go up to 20 or 30 layers of peptidoglycan, but much less than the gram-positive cells. Any question about any of that? Come on. Can't now move the slide. There we go. So the crystal violet iodine complex forms in all cells, both the gram-positive and the gram-negative. The crystal violet iodine complex is larger than the crystal violet or iodine alone. During the alcohol acetone wash in the gram-negative cells, the crystal violet iodine is washed out of the thinner peptidoglycan layer. The outer membrane of the cell wall is a lipid bilayer, and it's disrupted by the alcohol wash, so it'll be washed away. The cells will therefore be colorless after the decolorizing step of the gram-negative cells. In the gram-positive cells, the crystal violet iodine cannot be washed out of the thicker peptidoglycan layer. So therefore, the cells remain purple. Here we're looking at a gram stain, seeing gram positive rods, gram-negative spiral. There's a gram-negative rod. There's a gram-positive cockeye. And there's a vibrio, gram-negative vibrio. Any questions about any of that? Now, when you do the gram stain, you will often get results looking somewhat like that. And the question is, where do you read the gram stain? Do you read it here? Do you read it there? Do you read it there? Shrink that a little bit. These are the good regions of the slide. In fact, all of this region too. A good region is where the cells are a single layer thick, where the cells are not clumping. And let me blow this up. That's probably good enough. Where the cells are not clumping, and these actually are not clumps right there. This right here is a clump, but that right there is not. This is actually a crystal of crystal violet. The problem is that when you have your crystal violet solution in a container, it sometimes will form crystals of the crystal violet. And then those crystals will sediment usually to the bottom, but then you get your container of crystal violet and you put it upside down or whatever and squeeze out a bunch of crystal violet and the crystals come out. And then you got these crystals of crystal violet on the slide. You don't want to be reading the slide where there are clumps of cells or where there are crystals of crystal violet. And you can actually see this right here. If you notice, the cells are a bit darker right here and right there than right there or right there. This is especially a problem if you have gram-negative cells. These are gram-positive cells. But oftentimes on gram-negative, I'll notice that the cells away from the crystals are staining pink, as a gram-negative cell should. But then the cells right next to the crystal of crystal violet are actually staining purple. And the reason is that the crystal violet comes out of the crystal and then more darkly stains the nearby cells. And so you do not want to read a gram stain anywhere near a crystal of crystal violet. Any question about any of that? And then you don't want to read it also where there's clumping. I think I've mentioned this before, and that is with a clump of cells, it's hard to get the dye to go all the way through the outer layers of cells down into the inner layers of cells. And so they may not stain correctly. And then it's also harder to wash out the crystal violet iodine complex in a clump of cells, getting all of the stain out of the bottom layer of cells. And so the clumps just cause problems. You want to read the slide where the cells are a single layer of cells thick. Any question about any of that? When you do the gram stain, you'll notice that sometimes it feels like pulling out your hair. The gram stain can be tricky, and there are a number of things that can go wrong. The most likely things to go wrong is you do not apply the decolorizing agent for a long enough time, in which case the gram-negative cells may not all be pink after the gram stain. Some of them may stain purple because you didn't apply the decolorizing agent for long enough time. And then the other thing is, if you apply the decolorizing agent for too long, you'll pull out the crystal violet iodine complex from the gram-positive cells. Another problem is if you do not stain freshly grown cells. The gram stain only works well when you have cells that are 2 days old or younger. Anything older, you will have dead cells, and dead cells do not stain correctly. Their cell wall is deteriorating, and it just doesn't stain correctly on the gram stain. There are also some bacteria cells which stain poorly, if they stain at all, and that's mostly the mycobacteria, a family of bacteria that have mycolic acid in their cell walls. Mycolic acid is a lipid, and it makes anything that's in water resistant to getting across the cell wall because the water soluble molecule doesn't dissolve in the mycolic acid of the cell wall. The gram stain is using water-based dyes. The safranin is entirely in water. The crystal violet is mostly in water. I think it's like a 95% solution of water. It may be a 5% solution of alcohol. The point is that they usually do stain. All the mycobacteria I've used, at least if you use freshly grown cells, they do stain. However, their staining is going to be lighter because they stain poorly. And that's because the dyes do not get in the cells easily. They just don't dissolve in the mycolic acid. So the gram stain results are not always as clear-cut as theory suggests. When you're doing the unknown project, that may become apparent to you. The gram stain is not as clear-cut as it should be. All right, any question about that? Remember, if you're doing the gram stain, the cells have to be 2 days old or less. If they're older than 2 days old, you should streak out the cells and then do the gram stain on the freshly grown cells. Any question about that? All right. I went past that slide for some reason. The structure of the gram-positive and the gram-negative cell walls not only affects the staining, it also affects how they are related and how they respond to antibiotics. Like gram-positive cells tend to be easily killed by penicillin G, by cephalosporin, and many detergents. The gram-negative bacteria are more resistant to these antimicrobial agents. The reason being is penicillin G cannot get through the lipopolysaccharide layer of the outer membrane of the cell wall. And if the penicillin G cannot get into the cell, it cannot harm the cells. And that's true for cephalosporin and many of the detergents as well. Any question about that? All right. I need to talk a little bit about one stain, the special stains, particularly the capsule stain. You're not going to be tested on this, but in your unknown project, you may need to perform the capsule stain. This is a stain that is used to stain for the cell capsule. When you stain for the capsule stain, you stain the background of the cell, meaning the glass, and the cell. You do not stain the capsule that is around the cell. And that's best observed with the negative stain using India ink, which stains the background very dark. It lightly stains the cell, sort of a gray color, and then the capsule is unstained. Any question about any of that? All right, that's it for chapter 3. Let's move on to chapter 4. Any questions about chapter 3? All right, chapter 4. This slide is showing you the major goals and the rough outline. Know the terms of the chapter. What's a coccus, bacillus, spirilla? What's a strepto, like streptococci, staphylococci? What is that? Diplococci. What's a tetrad? What's a sarxinia? What's a vibrio? What's a spirochete? What's a glycocalyx? What is a flagella? Axial filaments, fimbriae, villi, peptidoglycan, endotoxin, selective permeability, diffusion, facilitated diffusion, osmosis, active transport, tonicity, what's a plasmid, inclusions, endospores, and the endosymbiotic theory. That reminds me, especially since we're behind schedule, on this lesson, I usually do discuss the functional anatomy of prokaryotic cells. However, the functional anatomy of eukaryotic cells is something you should have had in your preliminary biology course, which for most of you would have been Biology 160 at Clark College. So I will present the information in the PowerPoint, but I am not going to lecture over the functional anatomy of eukaryotic cells because it should be a review for you. I will, however, discuss the endosymbiotic theory, which are the last 3 or 4 slides of this PowerPoint. And that theory concerns eukaryotic cells, how eukaryotic cells obtain their mitochondria and their chloroplasts. So I will discuss that, but I will not talk about the functional anatomy of eukaryotic cells. Any question about any of that? And that reminds me, I just finished that lesson, so let me go ahead and make the PowerPoint available to you. All right. We'll also discuss how gram-positive and gram-negative cell walls differ, and we'll go into that in more detail. And then lastly, be able to describe the importance of taxis in bacteria. Any questions about what we're going to cover? If not, let's go to the first slide. When we're discussing prokaryotes, the term literally translates to pre for pro, and then kernel for nucleus. So prokaryote is a Greek word meaning pre-nucleus, literally pre-kernel. Okay. Eukaryote is a Greek word for true nucleus. Literally, that would be true kernel, but we're using the word kernel for nucleus. Prokaryotes in general have one circular chromosome. It's in a circle where the beginning point of the circle is the same point as the ending point. This chromosome is not in a nucleus. That differs from eukaryotes, where generally they have several to many linear chromosomes. An X chromosome is a linear chromosome, and it has a starting position and an ending position, which are not in the same place. It's not a circle. And their chromosomes are in the nucleus. The DNA of prokaryotes is not associated with the histone. The DNA of eukaryotes is. The histone is a large protein, and the DNA actually wraps around it. Prokaryotes do not have membrane-bound organelles like the chloroplast or the Golgi complex. Eukaryotes do have membrane-bound organelles. Peptidoglycan is found in the cell walls of bacteria, and only bacteria, not for the archaea. And there is no peptidoglycan in the cell walls of eukaryotes. Animal cells do not even have cell walls. Prokaryote cells mostly divide by binary fission, where the cell literally splits in two. Eukaryote cells divide by mitosis. Any question about any of that? All right, so the chapter summary, we're going to talk about prokaryote cell shape and arrangement. We'll talk about cell structures, and then the movement of material across the cell membranes. The eukaryotes, we do have lecture slides on the cell structure of eukaryotes, and it's in the slide presentation, but I'm not going to cover that. I will cover the endosymbiotic theory of eukaryotes. Okay, prokaryote cell size and shapes. The average size of a prokaryote, it's actually the average cell would be about 1 to 2 micrometers, but they're saying the average here is from 1 to 10 micrometers. The prokaryotes basically have three main shapes. There are round cells like spheres that are called coccus. There are rods, which are called bacillus. And it is true that you may call a bacillus a rod, that both of those terms are used interchangeably. And then there are spiral cells, shown here. There are different types of spirals. There's the spirillum, which is just an S shape, and it's a rigid spiral corkscrew shape in the shape of a letter S. There's a half a spiral, and you can call this a bent rod as opposed to half a spiral, and that's a vibrio, a bent rod or a half a spiral. And you could call that a spiral or half a spiral, or you could call it a rod, either way. And then there's a spirochete, and that's a spiral with many spirals. The spirochete is more flexible than the spirillum. This cell can literally bend. And I don't remember if I've shown you that movie or not, but the spirochete can actually, hopefully you can see this. It can actually bend on itself like this. And that's how flexible it is. Any question about any of that? There are a few prokaryotes that have unusual shapes, like the Stella bacteria have a star-like shape, five-sided star. This is the actual electron microscopic picture. This is the artist's rendition. And then there's the archaea, haloarcula, which this says square, but if I look at that, I'd say that's a rectangle. And that's actually a electron microscopic image. Most bacteria do not take on these shapes. Most of them are either cocci, bacillus, or spirals. Most bacteria have a single shape where all of the cells will have the same shape, like the haloarcula, they're all rectangles. But there are a few species. Oh, I guess we should mention the name there. If the species has a single shape, we call that monomorphic, where all the cells of that species have one single shape. But there are a few species of bacteria which are pleomorphic, where the cells of the species take on different shapes. Most of the species of bacteria, which are pleomorphic, are species that do not have a cell wall. And that's why they become pleomorphic. And that's because the cell membrane just doesn't give it shape. And the cell wall does, and these cells do not have a cell wall. So they take on many different shapes. We can also, well, wait a minute, let's go on. While we're talking about cell shapes, there is one cell shape in between a coccus and a bacillus, and that is called the coccobacillus, where cells, let's blow that up even further. The cells are in between a rod and a coccus. See that cell there? It looks like a coccus, but all the others are coccobacillus. That one, if you extend it, that one would be a bacilli, but that's because it's about to divide. That one almost looks like a bacillus too. What happens if you shrink the size of the bacilli right here? And if the bacilli has a round edge and you shrink it, it'll eventually become a coccobacillus. And this cell is in between the shape of a coccus and a bacillus. Any question about any of that? Let's go down. There we go. So we can talk about the prokaryote cell arrangement. This is also called the cellular growth arrangement. And that's how the cells stick together. When the cells divide, sometimes they stick together, and it's the cell wall that is sticking together. You can have singlets, like that cell right there is a single cocci, not stuck to any other cells. And then you can have diplococci. Let me blow this up. Where you have two cocci stuck together. And that would be a diplococci. That also is a diplococci. You can have streptococci where the cocci are stuck together in a row. And there's an actual electron microscopic image of a streptococci. You can have cells that take on the tetrad shape, 4 cells stuck together. A sarcenia, 8 cells stuck together. And you can't see the 8th cell because it's back here. And then with the cocci, you can also have a clump of cocci stuck together. And we call that a staphylococci. Staphylo means literally a cluster of cocci that looks like a grape cluster. So staphylo means grape-like, and cocci is obviously the shape of the cells. So if you put a stem on right here, and that would be a cluster of grapes. Any question about that? The cocci have more cellular growth arrangements than the bacilli. So how many do they have? 1, 2, 3, 4, 5, 6 different cellular arrangements. For the bacilli, they also have cellular growth arrangements. You can have a single bacillus. That's right there. You can have diplobacilli, where the 2 bacilli are stuck together. I don't see that in that picture, but that's okay. You can have streptobacilli, where the bacilli are stuck together lengthwise. So come on, mouse. Where the cells are linked together, sort of like some sausages hooked together like that. If you've ever seen sausages like that, you know what I'm talking about. You can have bacilli stuck together along the long edge, and there we're seeing it there, and that's called a palisade. Let me open up paint here. If you ever read Mark Twain, Tom Sawyer was painting a fence that was called a palisade fence. And the boards of wood are stacked like that, the soil being down here. And Tom Sawyer tried to convince Huckleberry Finn that painting the fence was fun. And so Huckleberry Finn started painting it, and then Huckleberry Finn did the work that Tom Sawyer was supposed to be doing, the painting of the fence. Anyways, that's the palisade fence, where the fences like that, and the cells reminded people of a palisade fence, and that's why it's called a palisade. Any question about that? The coccobacillus should not even be in this slide because this is a slide of the cellular growth arrangements, and that's not a cellular growth arrangement, that's a cell shape. When we're discussing the word bacillus, you should realize it has two different meanings. If the word bacillus has a lowercase b, that means we're referring to the shape of the cell, a bacillus. However, if the B has a capital letter, that means the family of bacteria that is called bacillus. Any questions about that? So pay attention to the capitalization, meaning the family of bacteria, lowercase b, meaning the shape of bacteria, and then don't get confused with streptococcus. That is a family of bacteria. Don't get it confused with, where do I have that? Streptococci, which is the cellular growth arrangement of certain cocci. Any question about any of that? All right, so the cocci have more cellular growth arrangements than the bacilli. The spirochetes actually don't have any cellular growth arrangement except for singlets. All of the spirals are singlets. They do not stick together when they divide. Any questions about any of that? All right. If not, let's talk about the structure of typical prokaryotic cells. We're looking at the structure here. The structures in red are structures found in all bacteria. So all bacteria have a cytoplasm, they all have ribosomes, they all have the plasma membrane, and then they all have a nucleoid region. The nucleoid region is the cytoplasm where the DNA is in solution. There is no distinction between the nucleoid region and the cytoplasm. It is wherever the DNA moves to. The DNA is in solution in the cytoplasm, and wherever it's floating around, that is called the nucleoid region. It does stain differently, the nucleoid region, from the rest of the cytoplasm, but there is no membrane in between the DNA and the cytoplasm. Any questions about any of that? All right, we'll talk about each of these typical prokaryotic structures. You find the pilus on a few species, not most species, but actually only a few. Fimbriae is found on more species. I don't know how many, if it's the majority of species or not, I don't know. The capsule is found on a lot of species, but is actually a minority of the species that have a cell capsule. Cell wall is found on most bacteria, but not all bacteria. Plasmids are found in many bacteria, and I don't know if that's most or not. Plasmids are a little different, and that is even within a species, you can have cells that have a plasmid and then other cells that do not. A plasmid is a small region of DNA. And then there's flagella. The majority of bacteria do not have flagella. However, if the bacteria can swim or move, then it has flagella, because that's the only structure that bacteria have for locomotion. Any question about any of that? If not, let's talk about the glycocalyx. The glycocalyx is secreted by many prokaryotes, but not all. It is outside the cell wall, so we call it a structure that is outside the cell. There are 3 types of glycocalyxes, and that can be pronounced glycocalyx as well as glycocalyx. They're usually sticky, and there are 3 types. There's the capsule, which is a neatly organized structure that you can see under the light microscope. So if you can see it under the light microscope, it is a capsule. The capsule can help prevent phagocytosis of the bacteria by white blood cells. If the glycocalyx cannot be seen under the light microscope, and you have to use an electron microscope to see it, and if it's loosely organized or unorganized, and it's mostly water, we call it the slime layer. You cannot see the slime layer under the light microscope. And then there's a third glycocalyx. You find this glycocalyx in bacteria that are in a biofilm. We call it the extracellular polysaccharide. What happens is bacteria secrete the biofilm. And what this biofilm is the extracellular polysaccharide from all of the bacteria. And the extracellular polysaccharide makes the biofilm. We'll talk a little bit about the biofilm later in the term. Any question about any of that? All right. The flagella is a structure that cells have for locomotion. If the cell can move, it has at least one flagellum or maybe several flagella. The flagella is mostly outside the cell wall, but there's a little bit of the flagella inside the cell wall and a tiny bit of the flagella even in the plasma membrane. Flagella are made of protein that we call flagellin. There are 4 basic arrangements of bacteria flagella. You do not need to know this for quizzes and exams. You do need to know it for one of the lab questions. There's monotrichous, where the flagella are on one side of the cell. Amphitrichous, the flagella are on both sides of the cell, but a single flagella. And then there's lophotrichous, where they're on both sides of the flagella, both sides of the cell, but there are multiple on both sides of the cell. And then there's peritrichous, where the flagellum are coming out all over the cell. So it has many flagellum, and they're all over the cell. Flagella cause the cells to swim, run, or tumble. So if the cell moves, it has flagella. Taxis is the movement toward or away from a stimuli. There's 2 types of taxis. There's chemotaxis, where a chemical provides the taxis. And then there's phototaxis, where the cells are either attracted to light source or they're repulsed from a light source. and these cells are moving away from the light source. They're moving this way. There is a special flagella that we find in spirochetes. It is called the axial filament or the endoflagella. I use the term endoflagella. The endoflagella is anchored at one end of the cell in the cell wall, and then it wraps around the cell, sort of like, I don't know, a string wrapped around something. When you look at this, let me blow this up. That endoflagella is actually inside the cell wall. So the light pink is the cell wall, and it surrounds the axial filament, the endoflagella, which is the reddish pink. And so that endoflagella is inside the cell wall. Any questions about that? The endoflagella causes the cell to have a characteristic corkscrew-like movement. You'll have to watch the commercial to get this. And there you can see how fluid the movement of that spirochete is. Get out of the way here. How the cell actually bent on itself and then changed, moving from this way to that way to moving to this way to that way. Any questions about that? That movement we call corkscrew movement.
So in this video, we're going to cover chapter 3.
All right. It's thought that the spirochetes, with their corkscrew movement, can actually burrow through an intact mucous membrane. The fimbriae are shorter, straighter, and thinner than flagella, and they're not used for swimming. They're used for attaching to something. Some human pathogens have fimbriae, and they attach to human cells by their fimbriae. And that's shown in yellow here, the fimbriae. The pili are longer than the fimbriae, and they're usually only one in a cell. And let me blow this up. So in this cell, you can actually see several fimbriae here, and there's the sex pilus, longer, and I don't know, bigger too. It is used let me move that down. It is used to allow a plasmid to leave this cell, and the DNA leaves the cell and then can move into that cell. That's what the sex pili is used for. Used to transfer DNA from one cell to another. And that plasmid will copy itself, so only a copy of it sent into that cell. So this cell doesn't lose the plasmid. Any questions about that? We'll talk more about the sex pili when we talk about genetics of microorganisms. Oops, why am I there? Okay, the prokaryotic cell wall is complex, a semi-rigid cell structure that gives the cell its shape. So if a bacteria has a shape, it's because the bacteria, I should say one shape, it's because the bacteria has a cell wall. The cell wall can protect the cell and prevent osmotic lysis. The cell wall is located outside the cell membrane. The cell wall can contribute to the pathogenicity of some species. The cell wall is mostly made up of peptidoglycan, at least in bacteria. That's not the case for archaea. Peptidoglycan is a polymer of two disaccharide repeating disaccharides, NAG and NAM. NAG stands for N-acetylglucosamine, NAM for N-acetylmuramic acid. And you can learn, oops, no, didn't mean to do that. I'm trying to blow this up. You can learn them either by the name, the long name, or NAG and NAM. Most people go with NAG and NAM. And NAG and NAM in peptidoglycan is just one repeating sugar followed by the next, followed by the next. And we have a row of it. And then in the cell wall, we have multiple rows next to it, and then layers of rows or planes underneath this plane of NAG and NAM. And this makes the layers of peptidoglycan. So like I said, in the book, it shows you a gram-negative cell only being one layer thick of peptidoglycan. In reality, it's at least 3 layers. So the disaccharide links the polypeptides. The disaccharide holds the peptidoglycan together, but peptidoglycan is more than just the NAG and NAM sugar. There are also short peptides linking the NAG and NAM lattices together. And as you can see, this short peptide links that sugar to that sugar, linking this row to that row. And then we have another short peptide which links that peptide together to that peptide. And then that links this row of NAG and NAM to that row. And this is where peptidoglycan gets its name. Glycan stands for sugars, and that's the NAG and NAM, the sugars. And then the peptide from the peptide from the short polypeptides holding the NAG and NAM together. And that means peptidoglycan. Penicillin actually inhibits the lattice formation by the polypeptides. So penicillin could get in right here, and then this short polypeptide cannot be put in there. And so when the cell wall is being formed, and this polypeptide has not been put in yet, the penicillin can get in right there and then block the linking of this part of the cell wall to that part of the cell wall, and that can put a hole in the cell wall, and the cell membrane can protrude out of that hole. And then as water moves into the cell, that cell membrane can expand out of that hole, and it will just continue to expand until the cell membrane pops, and then that can cause lysis of the cell. And that's actually how penicillin kills the cell. It puts a hole in the cell wall. The cell membrane can protrude out of that hole. The cell membrane can expand as water moves into the cell, and eventually the cell membrane will pop, and that will kill the cell. Any question about any of that? All right, let's take a look at the gram-positive bacteria cell wall. It has the cell membrane at the bottom of it, and then layer upon layer of peptidoglycan. There is teichoic acid and lipoteichoic acid running through the peptidoglycan, and they help hold the peptidoglycan together, the layers of peptidoglycan together. It is thought that cations can move down the teichoic acid and then use the teichoic acid to move into the cell, the gram-positive cell. However, the cations can go right through the cell wall, so they do not require the teichoic acid, but it does help them move through the cell wall and then enter the cell. The polysaccharides in the cell wall can provide antigenic variation, giving the cells different antigens. The lipoteichoic acid binds through the cell wall and actually will link into the cell membrane. The lipid part, the lipoteichoic acid, is in the cell membrane. Come on, mouse, right there. Any question about any of that? All right. If there's no questions, I'm going to take a break and I will be back in about 10 minutes. Any questions about that? So you're going to get a 10-minute break today. All right. Oh my God. No, I was not. Now I'm recording. Thank you. Let's now move on to the gram-negative bacteria cell wall. It is a little different than the gram-positive cell wall in that it, has the cell membrane, which is a lipid bilayer on this side of the cell wall. It is a very thin peptidoglycan layer, which here it's shown as only one layer thick. In reality, like I said, it's at least three. And then it has another lipid bilayer on top of the peptidoglycan. This lipid bilayer is called the outer membrane of the cell wall. What do I want to say about that? There are a few other different molecules in the cell wall of a gram-negative cell. First of all, we don't have any teichoic acid running through the peptidoglycan. And then we have a lipid bilayer outside the cell wall. Let me blow this up. This bilayer of the outer membrane of the cell wall is the same as this bilayer and that bilayer, meaning the main molecule for the cell membrane is the phospholipid, and that's the main molecule for the lower layer of the outer membrane, of the bilayer in the outer membrane. And so it's mostly phospholipid. Now, this layer of the outer membrane of the cell wall does have phospholipid in it, and it's probably the main molecule. However, it does have other molecules in it, mainly lipid A and an O polysaccharide attached to it, making a lipopolysaccharide. Inside the outer membrane of the cell wall, we also have a porin protein, and the porin protein creates a channel, and any molecules that the cell wants to come in through the porin protein channel. And then any molecule that the cell wants to leave the cell leaves through the porin protein channel, meaning this lipid bilayer does regulate the movement of material into and out of the cell, similar to but different from the cell membrane. And it's the porin protein which is allowing many molecules to get through the outer membrane of the cell wall. In the cell membrane, we have a transport protein, which allows movement of molecules across the cell membrane. And there will be one transport protein for one molecule. So like glucose, for example, has a different transport protein than fructose. That's different, and that is the porin protein. Instead of allowing one molecule to move across, it allows a whole slew of molecules to move across. The reason why you need a protein to allow it is that most molecules cannot cross the lipid bilayer of the outer membrane or the cell membrane. The only way they could do that is if they're lipid soluble, or they're a very small nonpolar molecule like oxygen that can diffuse right through the lipid bilayer. And then things like fats that can move through the lipid bilayer, but most molecules cannot, like most carbohydrates, most proteins, most nucleic acids. And if you think about our cells like a blood cell, you're not going to have a whole lot of lipid floating around in your blood because the blood is mostly water. There will be some, but like I said, most of it will not be. And so most molecules crossing the outer membrane come through the porin protein. And then to cross the cell membrane, they come through a transport protein. Let's see. Let's talk a little bit more about this lipopolysaccharide. It's made up of two molecules attached together. Lipid A, the big blue one there, and then the hair-like structure, which is the O polysaccharide. Lipid A is the important molecule in the outer membrane because lipid A is an endotoxin. A toxin is something that is toxic to us, and lipid A is an endotoxin. Any question about any of that? Let me see if there's anything else I need to talk about here. Oh, you can have a lipoprotein in the cell wall of the gram-negative cell, and the lipid portion will either be in the outer membrane or in the cell membrane. And then the protein portion will be in the peptidoglycan part of the cell wall. All right. Any question about the gram-negative cell wall? Let me shrink that down a little. So the gram-negative outer membrane contains lipopolysaccharides, lipoproteins, and phospholipids. There is a region of it called the periplasm. That's the space between the outer membrane and the plasma membrane. The periplasm is where the peptidoglycan is. Oh, I thought I had that. Oh, there it is right there. The periplasm is right here. The region in between the outer membrane and the cell membrane. The gram-negative outer membrane can help protect the gram-negative cell from complement proteins, and antibiotics like penicillin G cannot get through the outer membrane of the cell wall. Complement proteins are a protein that animals make to help defend them, like humans have complement proteins that can attack bacteria, for example, and the complement protein has trouble getting through the outer membrane to attack a gram-negative cell. The outer membrane also has O polysaccharides. This is an antigen and can give the cell certain properties. Like, for example, when we say E. coli O157H7, this is a human pathogen, not a normal strain of E. coli, but a human pathogenic E. coli. O157H7, O157 is an antigen, and that's the O polysaccharide antigen. H7 is another antigen. And then I talked about lipid A being an endotoxin and porins making channels through the outer membrane. So when we look at the gram-positive cell wall and compare it to the gram-negative cell wall, the gram-positive cell wall has thick peptidoglycan, the gram-negative thin peptidoglycan. The gram-positive cell wall, the cell wall is a rigid structure. It is rigid in the gram-negative cell, but it is not as rigid as the gram-positive cell. The gram-positive cell has teichoic acid in it. Gram-negative does not. The gram-positive does not have an outer membrane. The gram-negative cell has an outer membrane. And in that outer membrane, we have lipid A, which is an endotoxin. Any question about any of that? All right. I think I'm going to stop here and I will continue with this in our next class. I will be in the lab at the normal lab time, 6:30, but only for 15 minutes to answer questions. If you don't have any questions, you do not need to show up. If you have questions about one of the lab projects, come to the lab and I'll answer your questions. Are there any questions before I go? All right, I will see you. Bye.